So what I thought I would do is I would cover a little bit of the radiopharmaceuticals and then move into some of the newer things. And this is the challenge I think that we've been a little bit beating around the bush on. But when you molecularly characterize this disease as the Stand Up to Cancer group did, what you figure out is that yes, there's AR, but there's probably more to the story. And the AR addiction needs to somehow be dealt with, but there are a variety of other alterations that are present, and many of these are currently undruggable. We just talked about P53, for instance, as being a currently undruggable target, and it's really complex. So the challenge of precision medicine is not only to identify the alteration, but also to potentially intervene in some sort of successful manner. And it turns out that there's a lot of heterogeneity and there's a lot of undruggable targets. So I just kind of put up what I'll call the challenge of treating advanced prostate cancer is yes, that AR addiction that Bill pointed out, but how do we target these cancers that are highly heterogeneous in genotype? How do we target cancers driven by multiple undruggable targets? So all we have to do is answer these simple questions, and then we're like off to the races, right? So it's pretty simple. Um, I had actually gotten interested. I think I'll tell the story briefly because it's probably a matter of frustration. Um, the year I think is 1991 or thereabouts, and I'm at the National Cancer Institute. I'm starting to do prostate cancer. We don't have anything. We had Sermon. That was our gift to the world was Sermon, and Sermon really wasn't a very good gift. It was pretty frustrating. And I'm cruising around and I meet this guy named Bill Geckler. And Bill Geckler is using something called samarium-153 to target bony metastasis. I said, well, Bill, that might work. And got involved with some of the radiopharmaceuticals relatively early on, and consequently became interested in radium. And it's pretty fascinating when you can target radiation in the right spot because radiation will kill tumors regardless of the underlying genotype. And this is kind of an example here. And the good news is that with a lot of hard work on a lot of people's part, and I really would like to call out two people that don't really get, people may associate Chris or I with some success of radium. That's not really true. The real accolades in my mind go to a guy named Roy Larson and Oyvind Brulik, who were a couple of guys who put together a company called Algeta. And Oyvind was an academic at the University of Oslo, and Roy Larson was living on the savings from his postdoc, right, writing patents and trying to put together a company. And so it started with just two guys in a room who managed to put radium together. And then, of course, the clinical investigators followed, but we didn't really deserve the accolades they did. So from the phase 3 called Alcemca, we learned a lot, and we didn't learn much. I think radium was probably safer than expected, associated with improvement in overall survival. We learned a variety of concomitant therapies for fun. We had external beam, we had bisphosphonates, we had all these old hormone agents, bicalutamide, dexamethasone, DES, all sort of mixed in, and that was good. I mean, you get an FDA approval, you get a lead article in the New England Journal. This is good stuff. But there are a lot of things we didn't know. There was no AbbVie, no Enza, no denosumab, no imaging of note, no analysis of chemotherapy after radium. This whole issue that we were just talking about the post-protocol therapies, we just really didn't have much on that. We had radium dosings are arbitrarily limited to 6 cycles. That was just a shot in the dark. Came out of a London hotel room where we said 4 seemed to be pretty good, 8 seemed to be a lot, so 6 must be right. So 6 it was. And we just had a whole lot more to learn. So among the important things that we wanted to learn was the question of whether or not if you took AbbVie and Enza, which I think are fabulous additions to our armamentarium, have shown real value through 302 and Prevail to be able to prolong survival and benefit patients. The question is if you added radium in, it would be better. And these were put together in randomized trials. And the ERA-223 trial, which is the AbbVie trial, was stopped early for safety reasons. And it was like, when I heard it was stopped early, I was thinking, oh, good, it's a positive trial. And I just didn't anticipate it was stopped early because it was a negative trial. And it wasn't just negative, it was bad negative. There were fractures that were higher, unequivocally higher, and there were deaths that were higher. And this was like a real wake up call. And all of a sudden, the heads are spinning. It's like, why did this happen? What do we do about it? Now, piece three is still ongoing, and there are a lot of eyes on piece three right now. And this is Bertrand Tomball, Silke Gillison are heading that up, predominantly European centric, a few sites opening up. We've had delays because of registrational issues in France, etc. But I think it's going to work. But all the eyes are upon that trial to see how it works out. Now, one of the things that I think was being earlier touted as a potential positive is that when you give an active agent like Avirenza and you take these patients, and this is a little bit of a CT scan showing a kind of partially lytic, partially blastic lesion, you begin to actually heal these lesions, and you get what we call a flare. I actually don't like the term flare. It's a flare that's in the nomenclature. Chuck, you can change that. So flare is actually an improvement of the disease that has an uptake in the isotope that we use in bone scan imaging, which is the simple phosphonate that binds to this bony matrix. It has nothing to do with the tumor, right? It has everything to do with the tumor matrix, and a healing bone takes up more than a lytic bone. And the question is, if this is where radium binds, and there's data, the idea was maybe this was a good thing because you would target the radium to where it needed to be, but maybe it's a bad thing. So one of the questions is, when do you start the radium? At this sort of at the start of responding patients, well, we got piece three ongoing, we've got ERA shut down, and we have all these other sort of opportunities to explore. And as suffice to say, there's been a lot of discussion with Bayer on this topic, and it's tricky because they have regulatory issues to deal with. The European regulatory agencies have come out with certain pronouncements like, if you give Abby, you can't give radium. And that's not finalized, but it's really pretty tight. No concomitant use at all. So, one of the things, and I think all of us in the room know the limitations of radium. It only goes to bone, and that if you're trying to treat cancer, you're treating more than just cancer in the bone. You want to treat cancer regardless of its location. So, one of the things that has gotten a lot of attention, and we haven't talked a lot about the PSMA. We could be talking about fluciclovine, but PSMA is where the therapeutics have gone, and it turns out that PSMA is highly expressed in a variety of prostate cancers. We're still kind of looking at the full spectrum. A lot of this is in the early disease, and there's not a lot of heterogeneity. By the way, as you get later on in the disease, there's heterogeneity in the uptake that is seen by PSMA. You end up with tumors that are FDG positive, PSMA negative, seen on the CAT scan and other such things. So the heterogeneity does increase with time, but nevertheless, PSMA can be targeted. We have small molecules, antibodies, diabodies, many bodies. You can target this in a variety of ways. And the ones that have probably been the more common ones that I've discussed is particularly the PSMA 617. This was invented in Heidelberg. It went to the German subsidiary of a Japanese company called ABX. I thought it was dead in the water because the Japanese didn't want to sell it. And then interesting things occurred, and a little company called Indocyte has gotten in control of 617 and its destiny. And 617 has a nice chelate that you can put on things like lutetium and bismuth and actinium-225. And now it turns out that there's a variety of studies that have been done with 617 lutetium, predominantly coming out of Germany, but also out of Australia. You can see that these are pretty heavily treated patients, which you can't see from here, but some of the patients have pretty good responses. And right now, there's a phase 3 in planning with Indocyte that's going to be moving forward, and it will open this year. So I think that's really a nice thing to be able to move this forward in a quick manner. Now, there are other ones. Everybody probably knows that there's a variety of PSMA molecules. Another one came out of Hopkins, has gone through Progenics. got picked up by a company called AAA. AAA in turn sold to Novartis, and now Novartis has got control of the DCF PYL 1 and 2 series of compounds from Hopkins, Marty Pomper, and they're pushing it forward. But they're going to be doing phase ones initially because they don't have the experience with the therapeutic. Alphas are interesting. If you don't spend any time in this field, alphas are, 2 protons, 2 neutrons. They're really pretty destructive. They're great at causing DNA strand breaks. It'd be great to have them targeted as alpha emitters. And the guys leading the charge here have been in Heidelberg, and they've been using the actinium-225. Here's their most dramatic case. This is not typical of this dramatic. This is like, docetaxel, gotta love this. 50 cycles. I have no idea what they were doing. Chromustine and hyperthermia, abiraterone and enzalutamide, radium, abiraterone again, estramustine, PSA almost 3,000, kind of clearing with PSAs undetectable with actinium-225, PSMA 617. So that is, that sort of gets your attention, and it makes you think that there's more to do it. By the way, the bismuth-213 has been ongoing, and of all places, South Africa. Okay, quite interesting. In Pretoria, they've been able to pull it off with bismuth-213. Now, we're at the beginning of this. We're talking about monotherapy, right? Monotherapy is when we just give this, but we have so much diversity here. We have small molecules, we have antibodies, we have more. And, it was interesting you talking, Larry, about the zirconium. So zirconium has a longer half-life, and you match it up with a different half-life. You have to be able to match the half-life of the molecule with the half-life of the isotope to make it all come together. And, Bayer's now got a substantial antibody program, but then they may want small molecule programs. And there are interesting things that you can go to with, say, thorium as opposed to the bismuth or the actinium, and the supply sources of actinium. It gets complicated. But you can put all these things together. You can do combination. You use different types of alphas. You can certainly use different types of betas. You can think about hormones as sensitizing agents, and clearly they're going to be more in this area. Radiation DNA repair defects. So we had one guy who was a rather phenomenal radium responder. Turned out he had a biallelic BRCA2 deletion. He was germline positive. We finally got a piece of tumor on him, and he was biallelic BRCA2. He ended up with 18 cycles of radium, but this radium was like magic for him and kept going along. So DNA repair defects, obviously, what are you doing with radiation? You're damaging the DNA. You have a repair with a DNA repair defect. It could be interesting. You don't have to wait for nature to do the experiment. You can intervene with the experiment. You can do PARP inhibitors, or maybe ATR inhibitors. ATR is pretty cool as well. And there are even some ATM inhibitors, but you can use radiation to inhibit DNA repair. Radiation and high-dose testosterone. There's a nice article called Testosterone, the Frenemy, and it's showing the fact that high-dose testosterone can actually inhibit the DNA repair, much like low-dose testosterone or low testosterone can. So that's sort of interesting. Rob, to drive you crazy, I've got one of those patients with a pretty phenomenal response. Old and new radiosensitizers, the idea is that you're taking a deposition of a radionuclide, and then it's going to be pretty destructive right around where it deposits, but as it gets further away, everything is going to diminish by the R-squared rule. So it's further away, less hits. Maybe you can sensitize it a little bit, so that's going. And of course, I think everybody's aware that immunotherapy is potentially combinable with radiation in interesting ways. So, it gets to be, all of a sudden it gets to be a big field. And with a little bit of imagination, you can think of a lot of experiments. So that's kind of where we are. So thank you very much for the opportunity to present. Yep, Dan.
So, you brought up a very interesting point about the biallelic BRCA loss. Why has there not been a prospective study done looking at BRCA? Because you would think that with DNA repair as being the major mechanism for radium, that these would be the patients who tend to show PSA responses.
So, it's interesting. I have pushed that about 49 times with Bayer, and to try to get them to support some genotyping to be able to define, the winners and losers. Of course, they have no desire to do that. And so everything we've done, we've done on our own. Evan Yu from up in Seattle had written up a few little series, and these cases are anecdotal. There's an anecdote from Emmanuel Antonarakis, from myself, from Evan Yu. So they're out there, and I think it makes sense.
Do we know exactly the type of DNA damage that radium induces? Is it the same as is the type of DNA damage the same as external beam radiation?
Nope.
Is it both single strand, double strand?
It's double strand breaks, I think.
Predominantly double
Yeah,
strand.
but it's oh shoot, I have a slide of how it works, and I'm forgetting the term. We don't have a radiation oncologist here, but it's anyway. Never mind. Yeah, you're right.
So what happens with particles is they're really big and they'll go very far. So they only go on the mass of like feet. And there's a bright peak, probably the bright peak of it's true for all the heavy particles that travel. So you've got a bright peak with heavy particles and a very limited amount of space.
Yeah.
It's very destructive small things.
Exactly.
The physical term is linear.
But it's not repairable by homologous recombination. So
No,
so the
it's
BRCA2 patient who we think of for the PARP inhibitors, this would be just a different mechanism of DNA repair required to overcome this resistance.
Chuck, the only caveat there is not all the breaks are double strand. I mean,
Right.
you can, so with a beta, you predominate
Right.
on the single strand. On the alpha, you predominate on the double strand, but there's heterogeneity in the type of breaks. It's not all one, all other.
Right.
Okay. So anyway, you can create scenarios in your mind where you might get synergy.
Oliver, I have a patient who actually had a pretty phenomenal response to lutetium. So I think he had 4 cycles, and he went from a PSA of 3,000 down to 12, and everything sort of went quiet on his scans. But now I don't know what to do with him. So, he's, of course, had everything. What do you think about retreatment or switching agents? We're looking into actinium. He has some mild cytopenias, but none of the salivary gland issues that you would expect. So what do we do with these patients after we're done? Where do we go from here? And is there the possibility of retreatment?
If they're not progressing, well, you don't have to do much, right?
He is.
And if they progress in small spots, then I think you have the SPRT option. If you can PSMA scan, get an oligomet, SPRT, the oligomet, then that gets you into business. If there's a systemic relapse, and this gets a little pie in the sky, if they're travelable,
we're sending him to Germany.
Yeah, the Heidelberg guys do have the actinium potentially available.
That's the option I came up with too. I was hoping you might have a better one, but thank you.
Yeah, I can give you the contact information.
We're
if you
already
like.
set up, but thank you
Got
very
it.
much.
Yeah.
So we have a love-hate relationship with PSA as an intermediate endpoint, but why does PSA seem to go down with PSMA-based radioimmunoconjugates and not so much with radium? Is it an issue of micrometastatic extraosseous disease in patients treated with radium?
Yes, I'll stop you right there. The answer is yes. So one of the cool papers from Germany using radium and PSMA scanning was looking at the response on PSA versus non-response on PSA, and the PSMA scan could delineate the responders, non-responders. They called it the gatekeeper. I'm sorry, it was one of those guys from Germany with a name about that long that I can't remember. But I can show you the manuscript if you like. And what they showed was the people who had bone only were, of course, the best radium responders. But otherwise, there are tons of people with these little PSMA positive micromets, and that's probably where the PSA rise is coming